I'm Joe Graydon.
And I'm Terry Graydon. Welcome to this podcast of the People's Pharmacy.
You can find previous podcasts and more information on a range of health topics at peoplespharmacy.com. COVID-19 continues to infect and kill far too many. Will new pills from Merck and Pfizer change the course of the pandemic? This is the People's Pharmacy with Terry and Joe Graydon.
Our guest, Dr. Ralph Baric, is one of the world's leading experts on coronaviruses. He was involved in the development of antiviral medicines, including remdesivir and molnupiravir. He'll give us an insider's perspective on the new antiviral pills under review at the FDA.
Will we continue to need vaccine boosters every 6 months to prevent infection?
If the coronavirus is here to stay, how do we need to adapt as a society?
Coming up on the People's Pharmacy, the future of new pills for COVID-19.
In the People's Pharmacy Health Headlines, when COVID-19 first appeared late in 2019, it seemed there were just two outcomes for people who caught it. They died, or they got better. Nearly 2 years later, scientists are looking at outcomes with a lot more nuance. A recent study suggests that many people who had a positive COVID-19 test were still reporting some troublesome symptoms up to 10 months later. The scientists used a pre-existing database including 11 million patients of primary care practices in Great Britain. 200,000 of these people tested positive for SARS-CoV-2 during the pandemic. Follow-up surveys revealed that people who had been infected were 3 times as likely to have trouble sleeping as those who had not. Fatigue was 6 times more common among the previously infected during the follow-up period.
This week, Pfizer submitted its application to the FDA for emergency use authorization of its new oral antiviral drug, Paxlovid. This follows on the heels of Merck's request for authorization of its oral drug molnupiravir. British regulators have already approved the Merck pill under the name Lagevrio. Both drugs appear to reduce the chances of serious illness or death. To be effective, however, they need to be taken at the earliest possible stage of infection with COVID-19. Like Merck, Pfizer will be permitting generic manufacturers abroad to make and distribute its oral antiviral COVID pills at significantly reduced cost.
A few weeks ago, a study published in The Lancet indicated that a very old antidepressant called fluvoxamine helped reduce hospitalizations and deaths in COVID-19 patients. The randomized controlled trial was conducted in Brazil and involved nearly 1,500 people with COVID-19. Those taking fluvoxamine were less likely to be hospitalized or to die. Now a new study published in JAMA Network Open analyzed electronic health records of more than 83,000 people. Those who were taking a selective serotonin reuptake inhibitor, especially fluvoxamine or fluoxetine, were less likely to die from COVID-19. You may not recognize the generic name fluoxetine, but the brand name Prozac is a household word. The authors conclude that SSRI-type antidepressants may be associated with a lower risk of death from COVID-19.
There's growing evidence that drinking coffee or tea may be beneficial for the brain. The latest study comes from data collected from the UK Biobank. Over 360,000 volunteers responded to questions about their coffee and tea habits. During the follow-up decade, 5,000 people were diagnosed with dementia and 10,000 had a stroke. Analyzing the data showed that people who customarily consume 2 or 3 cups of coffee, as well as those who usually drank 3 or 4 cups of tea, were less likely to be among those with dementia or stroke. People who enjoyed both beverages, 4 to 6 cups combined in a day, were 28% less likely to develop dementia and 32% less likely to have a stroke than people who drank neither beverage regularly.
Another study involving coffee was presented at the American Heart Association Scientific Sessions. The CRAVE study recruited 100 middle-aged volunteers with no history of heart disease or rhythm problems. The protocol randomly assigned participants to drink coffee or avoid it. They alternated every other day. They wore a Fitbit to track activity and sleep. They also wore a continuous electrocardiogram monitor and a blood sugar tracking device. When they drank a cup of coffee, they pushed a button on a recording device. Coffee had no effect on the upper chamber of the heart. Atrial fibrillation is a serious arrhythmia that was not observed to be more frequent when people were drinking coffee. A common but not worrisome heart irregularity called premature ventricular contraction, or PVC, was more common on coffee-consuming days. Coffee consumption was associated with more steps and less time asleep. And that's the health news from the People's Pharmacy this week.
Welcome to the People's Pharmacy. I'm Joe Graydon.
And I'm Terry Graydon. We're nearly 2 years into a pandemic that's turned all of our lives upside down. It took a lot of people by surprise, including a lot of public health officials. But there have been warnings for years, especially from people who study viruses.
Our guest today has been studying coronaviruses in particular for decades. He's been described as the coronavirus hunter, and he was uniquely positioned to study SARS-CoV-2 as it emerged.
He and his colleagues were working on antiviral treatments long before COVID-19 turned into a pandemic. The injectable drug remdesivir was a product of that research. So was molnupiravir, which is being considered by the FDA as an oral treatment for COVID-19 infections. What can he tell us about the future of COVID-19?
For the answers to this and many other coronavirus-related questions, we turn to Dr. Ralph Baric. He's the William R. Keenan Jr. Distinguished Professor in the Department of Epidemiology and Professor in the Department of Microbiology and Immunology at the University of North Carolina at Chapel Hill. Dr. Baric is a recipient of the North Carolina Award for Science for 2020 in recognition of his work to develop treatments and vaccines for COVID-19. He's a Harvey Weaver Scholar from the National Multiple Sclerosis Society and an established investigator awardee from the American Heart Association. In addition, he's a World Technology Award finalist and a Fellow of the American Association for Microbiology. He spent the last 3 decades as a world leader in the study of coronaviruses and has contributed to the UNC Chapel Hill's world leadership in coronavirus research.
Welcome back to the People's Pharmacy, Dr. Ralph Baric.
Hi, Terry. It's a pleasure to be here again. I hope you're doing well and having a nice fall.
We certainly are.
Dr. Baric, we spoke with you, it's hard to believe, back in May of 2020, when COVID-19 was just in its early stages. Daily case reports were low, and effective vaccines, well, they were under development, but on the horizon and uncertain. It wasn't clear that we would ever have a vaccine, and there were no effective medications against the virus. Now, here we are, almost 2 years later, and 250 million people have been affected worldwide. 5 million have died. In the U.S., we're approaching 50 million cases with 750,000 deaths. Was any of this predictable?
Well, the Chinese health minister on and around January 25th, 2020, reported to the world that the virus was undergoing community spread and asymptomatic spread. And when you hear about a respiratory virus that is causing significant disease and chaos, it was flat out chaos in China, that can spread asymptomatically and in communities. That means that we are on the verge of a pandemic. So I would argue that was the first statement that I heard that indicated that we were facing a serious threat to global populations.
Dr. Baric, why is asymptomatic spread so important for that scenario?
The problem with asymptomatic spread for a respiratory virus is that you have silent transmission networks that are spreading out through the community. And this is occurring in the months at least in the US and also in China and Europe and elsewhere. It's occurring underneath an influenza season. And so you don't know, a lot of these patients are having very mild disease or asymptomatic disease. They don't know they're infected, or they think they're just infected with a minor respiratory pathogen that goes away in a couple of days. So they stay functional in the community, and they spread the virus to many other individuals. And the original virus that came out of Wuhan, typically one individual would spread the virus to 3 other individuals. And this RO rate would cause for rapid expansion of the virus in communities. Over time, for example, this has increased. The Delta variant is thought the RO, where the number of people infected from a Delta infected individual is about 7. And so the virus has adapted. It's become more fit at replication and transmission in human populations over time. And so these silent transmission networks, since not everyone is getting serious disease, spreads through the community and allows for massive numbers of individuals to become infected quite quickly, leading to the COVID-19 pandemic.
Dr. Baric, what has surprised you about SARS-CoV-2 in this pandemic?
Well, it's always a surprise when a virus emerges from nature to cause a global pandemic. Historically, the emerging coronaviruses, SARS coronavirus in 2002 and MERS coronavirus in 2012, didn't cause global pandemics. They didn't transmit as efficiently in human populations. And so I guess the first big surprise was that this COVID-19 strain seemed so efficient at transmission in patients who were asymptomatically infected.
Now, we have been on a roller coaster ride. I mean, it seems like at the moment in the U.S., when we're talking to you, cases, they kind of stalled. They're downward slope, and they may be back on the trajectory up again. Some countries have done really, really well. Other countries, especially Eastern Europe, are a total mess right now. What do you expect for the future?
Well, the key in any population right now is vaccination. We have effective vaccines that significantly reduce the chance that you'll be infected. If you are infected, the chance of having severe or life-threatening disease is reduced by much greater than 10%. And so if people get vaccinated, we will bring this pandemic under control in terms of severe life-threatening illness. Right now, we have vaccine hesitancy in different parts of the country. And that's resulted in this sort of plateau where we're having about 70,000 plus cases a day and about 1,200 deaths per day, depending on which group is doing the counting in the United States.
Now, Dr. Baric, in terms of the vaccines and vaccine hesitancy, some people worry that because the mRNA vaccines were developed so quickly, they were on a fast track, that might pose risks. As we look back on the data, how should we assess the benefits and the risks of these new vaccines?
Well, let me start off by saying the assumption that you made that these were made very quickly is false. So messenger RNA vaccine technology started in the 1990s, and there were significant problems with developing that technology. And over about a 20-year period of time, they figured out the basic problems, which is how to stabilize the messenger RNA, how to deliver the messenger RNA, and how to make sure that the protein that's made off of it is made in large quantities and for a fairly long period of time. That took 20 years of scientific development. And then starting around 2008 to 10, to 12, they started to actually begin to test mRNA-based vaccines against different viral pathogens like HIV and influenza. In our own lab, we started working with the NIH VRC around 2018, developing a mRNA-based vaccine for MERS coronavirus. MERS coronavirus at the time was the only emerging coronavirus that was spilling out from animal reservoirs into humans at a reproducible rate. And so it was the logical virus to try to develop a messenger RNA-based vaccine for. And as we had developed a variety of robust animal models of human disease, we tested those mRNA-based vaccines in young and aged animals against lethal MERS coronavirus challenge in the BSL-3 laboratory and showed that they were extremely effective, not only in young animals, but in the much more vulnerable aged animals, because aged populations, aged animals, aged humans mount much less robust immune responses than young individuals. And the shocking thing with these vaccines, the mRNA vaccines, is that they made equivalent protective immune responses irregardless of age. And so that data was quite robust. It was available beforehand that it worked for coronaviruses. And that led to mRNA-based vaccines being one of the vaccine platforms that the U.S. government was interested in pursuing when COVID-19 came on the scene in late 2019 and into 2020.
Dr. Baric, this mRNA technology that you say has actually been under development for years, will it lead to other better vaccines against perhaps influenza or maybe the common cold, or what about HIV?
Well, the answer is all of those pathogens are being explored with mRNA vaccine technology. At this point, we don't know the answer to those questions. But I would imagine that given the success of messenger RNA vaccines with the emerging coronavirus, SARS-2, that we will see its application to many other human pathogens in the near future.
You're listening to Dr. Ralph Baric. He's the William R. Keenan Jr. Distinguished Professor in the Department of Epidemiology and Professor in the Department of Microbiology and Immunology at the University of North Carolina at Chapel Hill. Dr. Baric has devoted 3 decades to the study of coronaviruses and has been dubbed the coronavirus hunter. He received the North Carolina Award for 2020 in recognition of his coronavirus work as it relates to COVID-19. In addition, he's a World Technology Award finalist and a Fellow of the American Association for Microbiology.
After the break, find out if we'll need to keep getting boosters for COVID.
Did the slow pace of vaccination increase our chance of encountering more dangerous mutations?
You'll hear about a collaboration between UNC and Duke researchers on an antibody that might work against many different coronaviruses.
Could we develop a universal vaccine against all coronaviruses?
We also discuss the new oral antiviral drugs Molnupiravir from Merck and Paxlovid from Pfizer.
You're listening to The People's Pharmacy with Joe and Terry Graydon.
This podcast is made possible in part by CocoVia, maker of high-potency cocoflavanol supplements. Cocoflavanols are among the most well-studied plant-based nutrients, backed by 20 years of scientific research.
CocoVia Cardio Health is available in capsules or powder, providing 500 milligrams of cocoflavanols daily. This supports better blood flow and vascular performance.
CocoVia also offers Memory Plus, supplements with 750 milligrams of cocoflavanols. This product is backed by 4 different clinical studies demonstrating significant improvement in several aspects of memory.
CocoVia flavanols are derived from real cocoa beans. They offer you all the benefits of chocolate without the sugar. More information at cocovia.com.
Welcome back to the People's Pharmacy. I'm Terry Graydon.
And I'm Joe Graydon. The People's Pharmacy is brought to you in part by Kaya Biotics, organic probiotic products developed in Germany and made in the USA. KAYABiotics.com.
Also by CocoVia, offering its cardio health product with 500 milligrams of cocoflavanols in powder and capsule form. More information at cocovia.com.
Today, we're discussing treatments for COVID-19. Both Merck and Pfizer have submitted emergency use authorization applications for their oral antiviral medicines. Merck's drug molnupiravir has been approved in the UK already and is being distributed under the brand name Lagevrio. The Pfizer drug, Paxlovid, works differently, but it's also a pill, which makes it much easier to take than the infusion of monoclonal antibodies.
To help us better understand such treatments and a new antibody discovery, we are talking with Dr. Ralph Baric. He is the William R. Keenan Jr. Distinguished Professor in the Department of Epidemiology and Professor in the Department of Microbiology and Immunology at the University of North Carolina at Chapel Hill.
Dr. Baric, we've seen that the vaccines are very effective. They do have people well protected against SARS-CoV-2. But the protection tends to fade, and as a consequence, people who are at high risk and older people have been getting boosters. I'm wondering, are we going to keep needing boosters as we go along?
That's a great question. So let me start at the beginning. So yes, it's clear that the current vaccines, just like virtually all vaccines, not all, but many vaccines, will induce a robust immune response, very good, and then over time it begins to wane, and that's a normal process associated with how the immune system works for many pathogens, and so that's not too surprising. Another issue was that typically, if you want to maximize the durability of your response, there's some data that argues that you need to have more time between the prime, the first administration of the vaccine, and the boost, the second administration of the vaccine. And the longer you wait in terms of a couple of months or 3 months, the longer the immune response lasts before it begins to wane. Now, in a pandemic setting, you don't have the luxury of waning 3 months because you could potentially have severe cases occurring in people that receive a single vaccine dose. And so they compressed the vaccine timelines a little bit early on, with about either 3 weeks for the Pfizer vaccine, I think, and 4 weeks for the Moderna vaccine. And so there's always been some recognition that we would require a boost. How many boosts might be required depends on the biology of the virus and how immunity wanes in human beings. And so let's talk about the variability of the virus itself, and does it change its coat? The answer is yes, it is changing its coat. And we've had 4 major variants of concern: alpha, beta, gamma, and delta. The delta wave, I think, was most memorable here in the United States. And there are specific changes that are occurring in these viruses that allow them to be more transmissible, potentially more virulent, and to potentially knock down the immune response against the original strain that was included in the vaccine. And it's not a lot. It's 2 to 4-fold, 2 to 5-fold. But if any kind of population receiving a vaccine, there's a bell-shaped curve, and there are people that have lower responses and higher responses. And so if a virus changes its coat enough to knock down your immune response by 2 to 4-fold, somebody at the lower end of the immune response now could become vulnerable. Someone at the higher end, they don't even notice it because they're still resistant. The second variable is how immunity works in the human lung. It has a tendency to be much more durable in the lower respiratory tract, where if you get infected, you're fighting, you can end up quite rapidly in a life-threatening situation. And that immunity in that region is much more long-lasting. In contrast, in the upper respiratory tract and in the nose and the oropharynx, immunity has a tendency to wane much more quickly. And so what would happen then is that people can become infected because they don't have protection in their nose, but the virus doesn't spread to the lower lung and cause severe disease. And that's what we're seeing in many of the people that have received the vaccine. There's a fivefold reduction in risk of infection, but a much more significant reduction in hospitalization or death. And that's just the way that the human immune system works and how viruses respond to immune responses in populations. So the big question is going to be, are we going to see more significant variants in the future that have changed their coat much more extensively that allow them to escape from the existing immune response? And we don't know if that will occur or not. We have no model to follow.
Dr. Baric, does the pace of vaccine or the failure to vaccinate increase the odds of more dangerous mutations?
Yes. So the COVID-19 virus has a sort of a base mutation rate. In other words, as it copies itself over and over again, it typically will introduce a couple of mutations per genome. And if you imagine that virus titers in a patient are hitting 10 to the 8th, 10 to the 9th, or 10 to the 10th, which is somewhere between, 1 billion to 10 billion viruses. That's a lot of mutations. And that allows them to evolve in response to herd immunity, because as they get transmitted from an unvaccinated person or a weakly vaccinated person or a person that has had a natural infection but a very low immune response, you're selecting for variants that escape that low level of immunity and or lack of immunity. And the combination of vaccinated and non-vaccinated and populations that have had natural infection immunity is a perfect scenario for the evolution of new variants with greater and greater antigenic change.
Dr. Baric, there was an announcement just recently about a Duke-UNC collaboration. Very exciting. I believe your laboratory was involved in this research, and the scientists announced that they have found an antibody that might work against SARS-2 as well as other coronaviruses. What can you tell us about that antibody?
Well, this is the second antibody that our group has been involved with in terms of identifying that is capable of neutralizing all of the known SARS-related coronaviruses. That includes not only the 2003 SARS strain, but the 2019 SARS strain, all the variants of concern that have emerged since 2020, and a large number of zoonotic SARS coronaviruses. Those are SARS-like viruses that are present in bat species. And so both of these antibodies are able to neutralize all of those viruses and protect animals from lethal challenge viruses in animal models of human disease. The most recent antibody is called DH1047. It was developed in collaboration with Bart Haynes's group at Duke University. David Martinez was the lead investigator from my lab who carried out the research. And this particular antibody binds to a novel site in the spike glycoprotein. That's the protein that projects out from the coronavirus particle that gives it a halo or like a corona around the virus particle. And it binds to that part of the spike glycoprotein that grabs onto a protein on our cells to allow the virus to infect the cells. And this particular antibody, which was pretty novel, binds to one particular site that is conserved across all these strains. And so it's able to neutralize all the different strains when the antibody is mixed with viruses. And it also protects animals once they get challenged. So it's a very novel finding, not only for a therapeutic antibody that you can use against future variants as they emerge and the current variants that are circulating, but it identifies a key target in the SARS-related virus spike glycoprotein that if you can drive a vaccine to make that antibody more efficiently, then you have a broad-based vaccine.
So when you say broad-based, do you mean like a universal vaccine against all of the potential coronaviruses that are out there?
That's the next dream. So a lot of virologists and coronavirologists feel that there are some regions on the spike glycoprotein that can protect from infection and are conserved across all coronaviruses. We haven't found them yet, but we think they're there. It's just very rare. It's a very rare targeted site. But once you find it, you can now design immunogens that it can be included in vaccine that present them much more efficiently. So right now, it's just the group 2B coronaviruses, the SARS-related viruses, that we have good reagents that where you can have a therapeutic antibody that could be put on a shelf to treat patients that are infected with, SARS coronavirus 3 or SARS coronavirus 4 or SARS coronavirus 2 variants of concern. But then it also is a signpost that says, here's a key target for vaccine design that you can then make a vaccine that would work against all of these high-risk SARS-related strains. And so
you mentioned a dream, and we actually now have not one, but 2 oral medications, molnupiravir from Merck Ridgeback, which I believe you were involved in, and a drug called Paxlovid from Pfizer. Molnupiravir is already on sale in the UK under the name Lagevrio. So I guess the first question is, how well do they work?
Well, you're right. We were actually the group here at UNC in collaboration with Mark Dennison's lab at Vanderbilt were the sort of the key researchers that demonstrated that molnupiravir was able to prevent SARS coronavirus 2 replication in culture and to protect animals from a lethal challenge with SARS coronavirus 2. And so all of that, and we also determined the mechanism of action of the drug. We showed it could work in primary cells from the upper and lower respiratory tract and demonstrated that it could also work against very, very distant coronaviruses. And so this is probably, molnupiravir should be considered a broad spectrum anti-coronavirus drug that would not only work against SARS-2, but potentially anything that might emerge in the future. And so we were involved in all of that research. That was in collaboration with Emory. The drug, which was originally developed at Emory for other purposes, was licensed to Ridgeback Biosciences, who then went and began and initiated both phase 1 and phase 2 clinical trials. Some of those phase 2 trials are about to be published. And then in larger phase 3 trials, the drug was shown to be extremely effective. It reduced the risk of death by over 50% and the risk of hospitalization of over 50%. I think in something like 750 volunteers, there were no deaths in anyone that had received the drug. And there were about 8 deaths, unfortunately, in people that had received placebo. So they halted the study immediately. And that's what's being taken forward to the FDA for emergency authorization use in the United States. But the beauty of this particular drug is it's an oral medication. And why is that critical?
Yes, why?
Yeah, because as soon as you get infected and you know you're sick and you get diagnosed, it's a pill. You can take it and you can go home and take it. The only other treatments required for, let's say, for a therapeutic antibody, it would have to be delivered by needle stick or IV administration. Or in the case of remdesivir, it had to be delivered by IV administration. And there was a tendency not to use the drug until people were hospitalized. And the longer you go from the time of clinical onset or you begin to see symptoms and the administration of the drug, the less effective the drug will be. And that's true for a therapeutic antibody or molnupiravir or the new drug from Pfizer.
Or pretty much anything you're going to take against, for example, a cold or the flu. It's going to work better if you take it as early as possible.
That's correct, because peak virus titers with COVID-19 are occurring either just before or about the time you start showing clinical symptoms. And so if you wait another 6 days before you administer drug, the virus is already on its way to being cleared from the host. It's done its damage. It's initiated all these immune inflammatory programs that can progress to death. It's done extensive damage to the lung by direct virus cell killing. And since it's already being cleared, the drug doesn't have a big impact. But if you give it early, you knock down almost all of those features. And so the common theme of both the Pfizer drug, which is also an oral medication, but it targets a different part of the virus, and molnupiravir, which targets virus replication and knocks it down significantly. The common theme is you give it early, you save lives. And the effectiveness is directly proportional to how early those drugs are given.
And now that we have over-the-counter tests for COVID-19, if someone takes the test, 15 minutes later is positive, they could be on the phone to their doctor. Their doctor could send in an electronic prescription for the drug, and they could be taking it within a day.
That's correct. Both of these drugs are game changers. It's always, the Achilles heel of remdesivir was always that it had to be given by intravenous administration. Now, I'll also just mention we have a paper in press with Gilead that's an oral version of remdesivir. So there's also an oral remdesivir that is being developed. And remdesivir is actually extremely effective if it's given early by IV. And in fact, if you look at the data for the people that received remdesivir as soon as possible after diagnosis and clinical onset, there's a 23 to a 54% reduction in mortality. So remdesivir has saved tremendous numbers of lives. We have just historically been using it too late. And, the first clinical trials that said remdesivir didn't work, they were given at day 10 or 8 to 10 post-symptomatic onset. The virus was already cleared. It wasn't going to do anything. Therapeutic antibodies don't work there either. You have to give them early.
You're listening to Dr. Ralph Baric, the William R. Keenan Jr. Distinguished Professor in the Department of Epidemiology and Professor in the Department of Microbiology and Immunology at the University of North Carolina at Chapel Hill. He's been studying coronaviruses for more than 3 decades.
After the break, learn about possible side effects from molnupiravir.
Might the antiviral drugs increase the possibility of future virus variants?
Will the virus develop resistance to these drugs?
What are the differences between Paxlovid and molnupiravir?
How are they likely to change the shape of the pandemic?
We'll also consider how society will need to change if we all need to adapt to SARS-CoV-2 over the long term.
What lessons should we take away from the COVID-19 pandemic that can be applied to the next pandemic?
You're listening to The People's Pharmacy with Joe and Terry Graydon.
This podcast is brought to you in part by Kaya Biotics, organic probiotic supplements. These supplements are made in the USA with high-quality, sustainably sourced ingredients.
Originally developed in Germany, Kaya Biotics offers 3 different formulations with 15 carefully selected strains of bacteria. These are designed to increase the diversity of your gut flora and support your immune system. More information at kayabiotics.com. Welcome back to The People's Pharmacy. I'm Terry Graydon.
And I'm Joe Graydon.
The People's Pharmacy is brought to you in part by CocoVia, now offering its cardio health powder with 500 milligrams of cocoa flavanols. More information at cocovia.com.
And by Kaya Biotics, microencapsulated, organic, hypoallergenic probiotic products developed in Germany and made in the USA. KAYA Biotics.com.
Our topic today is new treatments against COVID-19. How will the new antiviral pills change the pandemic? If SARS-CoV-2 is here to stay, how will we adapt?
Our guest is Dr. Ralph Baric. He's the William R. Keenan Jr. Distinguished Professor in the Department of Epidemiology and Professor in the Department of Microbiology and Immunology at the University of North Carolina at Chapel Hill. Dr. Baric has devoted 3 decades to the study of coronaviruses.
Dr. Baric, what about side effects? There have been some concerns raised about molnupiravir in particular, concerns about potential birth defects if a pregnant woman were to take it, or the possibility that it could increase somebody's risk for cancer. What do we know about side effects?
So you have to understand how molnupiravir works. And this is a drug that's called a nucleoside analog, and it's incorporated into the RNA genome of SARS-2 when it replicates. And it's considered an RNA mutagen. In other words, it mutates RNA that is rapidly replicating or rapidly being synthesized. And since the majority of RNA in a virus-infected cell that's being synthesized is the virus, it basically mutagenizes the virus and causes a phenomena called error catastrophe. In other words, when it introduces changes, it puts stop codons in proteins, it destroys regulatory regions in the virus, and it kills its ability to replicate. Now, molnupiravir is taken up into the cell, and its potential mutagenic effects are related to the fact that there are biosynthetic pathways in host cells that can take the molnupiravir drug and convert it into a DNA intermediate that can be taken up into DNA. And so if you, in a study that we did on looking at the mutagenic effects of the drug with Ron Swanstrom here at UNC, if you put high concentrations of drugs in cells in culture and let it sit for 30 days, again, this is at a length of period of time that's not used in human patients, you could see very low levels of mutations in host DNA. And so the drug can be metabolized, and some small fraction of it could be taken up into a rapidly growing cell. So a cell that's dividing rapidly might be able to have a mutation in it. Now, the companies have an in vitro, a cell culture model versus what happens in animals are very different things. And both Merck and Ridgeback have done a tremendous number of refined assays that are FDA standard assays to look at the mutagenic effects of a drug under the normal treatment course and dose in animals and in other cell culture systems. And they don't see a signal on this 5-day interval course that they use. So it's an issue. It's an issue that needs to be discussed at the FDA. But for most of us, the risk is probably exceptionally low. You probably are at greater risk being in sunshine, going to tanning beds, smoking cigarettes, drinking alcohol. What else? What else would be a good?
Well, there is another question that has come up, and that is by the nature of the mechanism where it introduces errors in replication of the virus, could it increase the possibility for future mutants, for variants that'll be even more dangerous, for example, than Delta?
The answer to that is we've looked at that to some extent. Typically, what's happening is many mutations are neutral, more mutations are deleterious, and advantageous mutations are rare. That's sort of a classic evolutionary theory. And so what molnupiravir does is that it introduces so many deleterious changes that the viruses that come out are highly attenuated. And the virus has to repair those changes. And there are multiple sites that it has to fix simultaneously. And so I think there's potential risk there, but I think it's relatively low risk.
Will the virus develop resistance?
So against the Pfizer drug, certainly. Absolutely, no question. Against molnupiravir, we have tried to do this. It is extremely difficult for the reason that I just told you. The drug is introducing so many other changes elsewhere in the genome that the virus is trying to compensate with second additional, they have to either revert those sites back or they have to put mutations elsewhere to compensate for the deleterious effect of the mutation. And so in the background of trying to select for resistance, you have all these other mutations occurring across the genome, and that's called lethal mutagenesis. It's just overwhelming for the virus. It can't survive it.
In the early studies that are being submitted to the FDA, the Paxlovid from Pfizer showed an 89% effectiveness versus a 50% effectiveness for molnupiravir. Does that give it a clinical advantage?
It's hard to say. The actual studies haven't been published yet, so I don't know how early the patients were being treated in the two studies. So, for example, if in general the population in the molnupiravir study is a day or so later before drug is administered than what's seen in the Pfizer study, then the Pfizer study is going to look better. If they are equal, all things equal, in terms of age and day of administration and time from clinical onset, then I would probably argue the Pfizer drug may have had advantages. But I don't think anybody can really make a solid statement on that until they know the actual details of the study.
Now, I have to admit, we were really impressed with Merck because the company says it's basically going to be giving away the patent information to low and moderate income countries, much as they did with ivermectin 20, 30 years ago when it was being released for river blindness in Africa. But it's going to be expensive here in the U.S. So far, it runs about $700 for a 5-day course of treatment. We don't have any idea how much Paxlovid is going to cost, but I'd just like to get your perspective. I know you're a scientist and not an economist, but the cost of this drug that could be, in fact, a game changer.
Well, it's absolutely a game changer. I mean, even there were initial discussions about remdesivir, which ran about $2,300 per treatment course when it first came out. But the savings in terms of hospital costs, time to release of the patients was about five days. And even when administered late, 5 days was a significant, a release from the hospital 5 days early was a significant financial savings to the patient and to the insurance companies. So there was a significant savings at that price. At $700, the impact is even going to be greater to the healthcare costs across the U.S. because most of these patients aren't going to end up in the hospital. It's not only a huge reduction in death, but it's a huge reduction in hospitalization and associated costs. And when you remove also the costs associated with intensive care, the savings at that price for the United States is a blessing. I think the fact that Merck is making the formula and the manufacturing process of molnupiravir to the world at large is a sign of a great company.
Dr. Baric, how do you expect these oral medications to change the shape of the pandemic?
Well, again, we are seeing about 1,200 deaths per day in the United States. Most of those are in non-vaccinated individuals. I think less than 100 are in vaccinated individuals. And the difference is going to be that those patients can go on life-saving oral medication earlier. And so that could reduce, again, if it reduces the mortality by 80%, we're talking about the impact of the drug reducing mortality from about 1,200 to about 200 to 300. And, that's a lot of people that get to spend time with their families and, for decades to come and to enjoy the holidays. And that's a game changer.
Dr. Baric, we have had lethal pandemics every few decades. I mean, in quote unquote recent memory, the 1918 flu, polio in the 40s and 50s, swine flu, AIDS, Ebola, and now COVID-19. When do you expect that we'll have another pandemic, perhaps something from a completely different virus? And how do we develop a pandemic preparedness plan that will actually be more proactive?
Yeah, so that's a great question. So let's just focus on the 21st century. There have been 13 epidemic or pandemic virus episodes in the 21st century. 3 emerging coronaviruses, and the 2009 influenza virus, the alpha virus, chikungunya virus, both the West Nile virus and Zika virus, 2 flaviviruses, 4 norovirus epidemic or pandemics. Noroviruses cause gastroenteritis disease. They only kill about 800 to 1,000 people each year in the United States, mostly young children. But globally, they cause 200,000 deaths each year in a non-pandemic year. So we live in a dangerous world full of pathogens that are capable of causing considerable morbidity and mortality to human populations, as well as disrupting the supply chains and the global economy. And so we do need to take a much more proactive response, as I just mentioned. And there's a couple of ways to do this. We know pretty high certainty what are the top, 9 to 10 virus families from which pathogens can emerge. We know where we sit in the current scheme in terms of antiviral drugs, immunotherapeutics, and vaccines, and how well they would be developed and how long it would take to get them developed. We know what the vulnerabilities are in which each of those virus families, a major vulnerability usually falling around the concept of animal models and available animal models that replicate the kind of disease that's seen in humans. It's easy to test a vaccine and show that it works against something where the virus barely grows in an animal, right? That's not what you want to test it against. You want to test it in an animal model that recapitulates the kind of disease seen in humans and the kind of replication that the virus is capable of achieving in humans. And so a lot of these different virus families, we don't have those kind of reagents, those kind of animal models available. And so that's a high priority area of research. A second area is that we can take prototype pathogens in each of those families and figure out, to a large extent, what the key issues might be in developing vaccines, what the key issues might be in developing therapeutic antibodies, and what the key issues might be in terms of developing drugs. And so investing in that kind of preparedness research for many of these virus families that have not received the kind of global public support through the NIH and NSF and other funding agencies in the United States or the WHO or globally, to invest into those pathogens so that we could be better prepared. To some extent, we were lucky with coronaviruses. We were lucky because before COVID-19 emerged, we had SARS coronavirus in 2002, which taught us a whole lot about what the barriers might be in terms of vaccine development and where the major targets of protective immunity are in the virus. And then we had MERS, which taught us even more. And so those acted as, in essence, instruments of instruction so that we could be better prepared and respond more quickly to COVID-19. We don't have that kind of information on many other virus families, and that's what's desperately needed.
Dr. Baric, some people have suggested that we might have to learn to live with this virus, that it may be with us not just for years, but perhaps for decades. If SARS-2 is here to stay, how do we need to adapt as a society to such an endemic infection?
Well, I do personally believe that we have been colonized. Humans have been colonized by SARS coronavirus 2, and it's not going to go away. And it's going to be the fifth human coronavirus that circulates endemically in human populations. Now, prior to SARS coronavirus, had either of you heard of a coronavirus?
Only in technical aspects, I think.
Well, we know that you've been studying coronaviruses for what now, almost 35 years, but the rest of the world was pretty oblivious.
That's right. And that's because the contemporary human coronaviruses typically infect you when you're very young. Most of us are infected with two or more strains before the age of 5. 90% of us, within a few years of that, infected with 3 or more of those strains. And coronaviruses in general don't cause lethal disease in extremely young individuals. So most of us have respiratory infections that we recover from. Now, there are examples of lethal infections with the contemporary human coronaviruses, but they're not very frequent. The beauty of that biology is that you get infected early and you get an immune response. But because of the way the immune system wanes in the upper and lower respiratory tract, every 3 to 5 years, you get reinfected with that same coronavirus. It replicates in the nose and the oral pharynx and typically doesn't go to the lower lung. Unless, you're immunocompromised or you have a really low immune response or you're an outlier, right? But for the vast majority of us, we get boosted every 3 to 5 years. And as our vulnerability to lethal infection increases with increasing age, we have been, in essence, vaccinated, revaccinated, vaccinated by natural infection many times before we're really in a highly vulnerable state. And so it isn't until you're in your late 70s and late 80s when your immune system begins to fall naturally just because of longevity and age that coronaviruses then reemerge as significant human pathogens and can cause mortality. The contemporary coronaviruses can cause mortality in older individuals. And so COVID-19, in my opinion, is going to recreate that scenario. We have a large number of people that's been vaccinated, but the vaccines only reduce the chance of reinfection by about 5-fold. But you don't die. The mortality rate has been significantly reduced because of vaccination, but you get boosted. Your immune system gets boosted. And then 5 years later, it gets reboosted. And so that's what I would imagine what's going to happen. It'll become that kind of scenario. There will be mortality associated with it, but it will be at a much lower level than the 2% that's occurring right now globally. It's going to be 0.01% or 0.001%. And we live with that kind of risk from respiratory viruses all the time.
Dr. Baric, what lessons should we take away from the COVID-19 pandemic to help us prepare for the next pandemic, whatever it might turn out to be?
Well, the response to COVID-19 is a triumph of science in the 21st century. In 10 months, we had an FDA-approved drug. In 11 months, we had an FDA-approved therapeutic antibody. In 12 months, we had a FDA-approved vaccine. That has never been done in the history of science. Even with all that success in terms of the rate at which we were able to develop countermeasures, we've still lost 5 million people globally and 750,000 in the United States. And that's because the response, for the most part, was reactive rather than proactive. We had data from prototype pathogens like MERS. We had data from prototype vaccines like mRNA-based vaccines. We had data on remdesivir and molnupiravir before COVID-19 emerged that those things could all work. And that's why we were able to do it so quickly. But the truth is, if we'd had another year or two, we would have had an oral remdesivir ready to go. molnupiravir had gone through phase 1 trials. And so it would have been better positioned to immediately go into phase 2 trials as the cases ramped up early in the pandemic. We would have had experimental vaccines that moved the bar from a type-specific vaccine against one pathogen to a broad coronavirus vaccine that would work against everything that we knew that existed in nature. And currently, our group we've published one on our own in collaboration with Duke and researchers at University of Washington. We've developed universal vaccines against all the coronaviruses, and we've shown they work. So we're much better prepared for future coronavirus outbreaks than we were 2 years earlier. But when we think about emerging diseases, these types of platforms have to be applied to about 9 different virus families to really put the world in a proactive position, ready to respond with things on a shelf so that we never have to go through this again.
Dr. Ralph Baric, thanks so much for talking with us on the People's Pharmacy today.
Pleasure talking with you both.
You've been listening to Dr. Ralph Baric. He's the William R. Keenan Jr. Distinguished Professor in the Department of Epidemiology and Professor in the Department of Microbiology and Immunology at the University of North Carolina at Chapel Hill. Dr. Baric received the 2020 North Carolina Award for Science in recognition of his work to develop treatments and vaccines for COVID-19. He's a Harvey Weaver Scholar from the National Multiple Sclerosis Society and an established investigator awardee from the American Heart Association. In addition, he's a World Technology Award finalist and a Fellow of the American Association for Microbiology.
Lynn Siegel produced today's show. Al Wodarski engineered. Dave Graydon edits our interviews. B.J. Liederman composed our theme music.
This show is a co-production of North Carolina Public Radio, WUNC, with the People's Pharmacy.
Today's show is number 1281. You can find it online at peoplespharmacy.com. You can subscribe to our free podcast through your favorite podcast provider. We post the show on our website on Monday morning. This week, the podcast contains information on how we can be proactive instead of reactive in facing a lethal pandemic. How will we need to change our behavior as a society? We just didn't have time for these topics right now.
If you'd like to add a comment about this show, look for the post on our website. You can enter your comment at the bottom of the show notes for 1281 under the podcast tab.
At peoplespharmacy.com, you can sign up for our free online newsletter to get the latest news about COVID-19 and other important health stories. By subscribing to our newsletter, you'll also get regular access to our weekly podcast and find out ahead of time which topics we'll be covering. In Durham, North Carolina, I'm Joe Graydon.
And I'm Terry Graydon. Thank you for listening. Please join us again next week.